Well, good afternoon. We'd like to thank the organizers for the kind invitation to address the conference. And as I said, that was a wonderful introduction to my talk. So I'm going to focus on the drivers of the obesity epidemic with an emphasis on the potential role of genetic differences and attempt to show what is the magnitude of the effect of the genes and how do they exert their influences. So if we take a mile high view of this, a very high altitude view of the drivers of obesity, they can be found in physics, physiology, behavior, and environment. And I think we can stop there because it encompasses everything that has the ability to influence body weight. Behavior, environment, we're rather familiar with those, and we are going to hear a lot about them in this conference. I don't think we're going to hear a lot about physics and physiology, so I'll say a couple of words on them. I seem to have a problem with this. Okay, so we've been told that energy cannot be created or destroyed, and in a closed system such as the human body, the heat supply to the system comes in the form of dietary calories, and the loss of heat from the system comes in the form of energy expenditure. Thus, if weight gain is going to occur, it has to occur in the presence of either an increase in caloric intake or a decrease in energy expenditure. That's what physics tells us, and that's what is the first law of thermodynamics. But that's not the whole picture. There are a few other things that come into play that influence the fate of the energy that is being consumed or expended. And physiology is what allows us to begin to understand these nuances in the fate of the energy currencies in the body. Processing metabolic fuel can vary depending on hormonal and on tissue and organ markers. For example, we know that insulin, glucocorticoids, leptin, steroids, and a few other growth hormones, other hormones, influence nutrient partitioning and the profile of the metabolic fuel, which goes for storage or for oxidation. Likewise, some tissues, major tissues like the liver, the muscle, and the adipose tissue, and their metabolic state influence the fate of how we handle metabolic fuels. So the laws of physics have to be seen through the prism of physiology. I'm going to present you now my comprehensive model, my global model of the causes of weight gain and the obesity epidemic. It's a little bit complicated, as you will see. This is what I call the internal system. Each of us has this system. Energy intake, energy expenditure, that's the physics component. Then we have the partitioning, the accretion, the adipogenesis, that's the physiology part. And then we have the outcome, either normal weight or when everything moves towards energy accretion, we have gain in weight, and eventually, if there's enough of it, obesity. But we know that this internal system is subjected to a lot of pressure from the social environment, from the physical environment, specific behavioral profile, and of course our own intrinsic biology outside of the one that I mentioned thus far. So that is a complex system. There's not one point of entry into this. There are multiple effectors of the regulation of the energy balance. But each of those four major categories of determinants can be also described in terms of multiple sets of particular effectors. And that's my version of it. And as you can see, and that's probably only a partial one, There's lots of factors in the social environment, the physical environment, behavioral, and particularly in biology. I have constrained the biology enormously here, which can vary between people and exert influences on at what level we regulate energy balance, the physics, and at what level the physiology comes into play to determine the specific outcome. Two of them, behavior and biology, are particularly relevant to the influence of genetic differences. So among the drivers, the genetic drivers will have its impact via its influence on biology and on behavior. Now, reorganizing this to make it simpler, we can pose that the built environment and the social environment, when they favor positive energy balance and fat accretion, they can be seen as defining what we have been calling an obesogenic environment. And when there is an obesogenic environment, that favors obesogenic behavior. And then this, in turn, impact on our internal individual energy balance regulators. But there is a biology that filters that. Some people have defects in their biology. For example, some people have no leptin. They have a complete deficiency of leptin. Their biology is defective, and of course they become, by adolescence, they have about 100 pounds of excess weight. And there are also others with less dramatic effects. So one question that one may ask is whether there is a genotype behavior interactions, typically known as the GE, genotype environment interaction. But in reality, here is genotype behavior. And a genotype behavior interaction effect is present when the response to a change in behavior is conditioned by the genotype of the individual. And that was something that was illustrated in the Drosophila in the prior talk. So here I'm going to show you two studies that have shown that there is a genotype behavior interactions in modulating body weight and adiposity. And there are more, but I'm going to show you two. One is a weight gain experiment. This is an overfeeding study that we did now 25 years ago. And here we have 12 pairs of identical twins. So therefore, each pair has the same genotype, exposed to an overfeeding treatment for 100 days. The overfeeding stimulus was an excess of 84,000 calories. So they had to buffer this. They were kept sedentary. And you have here on this x-axis the weight gain of one brother of each pair, and on the y-axis, the other brother. So one dot is a pair. So we have pairs that gain 5, 6, 7 kilograms, but we have pairs who also gain 11, 12 kilograms. Threefold range in the weight gain. And if you look at the within pair difference, you see that there was an F ratio of 3.4. So we have 3.5 times more variance between the pairs that we had between brothers in the same pair. A clear indication that there is a genotype overfeeding interaction effect. Another one is this one, where this is now a weight loss, 28 days in obese women, but they were female monozygotic twins. It was done in the Czech Republic. And again, you can see the same design of the graphs. After 28 days of weight loss, where they consume only about 400 calories a day, some lost a lot of weight and others less. A very strong genetically driven response. So because of that, then we need to modify my prior graph to allow for differences in the filter. Some have a strong filter, others have a very light filter. Some are really predisposed to favor positive energy balance and weight gain, and others much less. In addition, we have also other considerations. I'm not going to expand much on them, but we have maternal influences, and these are the nutritional state of the mother, obesity of the mother, the diabetes or the gestational diabetes in the mother, smoking and other behavior. And then we have in utero effects as well, like the wiring that's occurring in the development of the nervous system and the interactions with tissues and organs, hormone sensitivity, DNA methylation, and histone modification. All of this also can impact on that internal regulator that we have. So when we talk about the genetic component of obesity, then of course irritability is the key question. There are two types of irritability. What we should be interested in is what we call the narrow sense, but in practice we always use coefficients that represent the other one, the broad sense. It's a bigger coefficient, but it's not a more useful coefficient, it's just bigger. The very important one is the smaller one, and it is very hard to quantify in human populations. It's much easier to do in animal experiments. And the literature says that depending on the method that you use to calculate this irritability, it varies a lot, from 10 to 30% in adoption studies to 50 to 90% in twin studies. Obviously, some of them are wrong. We don't know which one. That will take a while until we have all the genetic variants and all the genetic features that are responsible for the irritability. We will be speculating as we've been doing for maybe 50 years. However, this is the aggregate trait, obesity. It's not very actionable as a measure. It's much more useful if we know the irritability component of the endophenotype, or the intermediary traits that are in the causal chain leading to obesity. There is a sample of those here from a number of family studies, and we see that they're lower. They tend to be around 20, 25%, as opposed in the 50, 60, 70%. Okay, now, what do we know about the genes now? Well, there have been genomic screens, and they continue to occur. A lot of genomic screens are in progress. A big one will be soon published. And the current number is about 100 genes that have been identified. It's more variants, DNA variants rather than genes, but we tend to refer to them as the closest gene or the gene within which the variants exist. If we take this illustration to show how these variants impact obesity, that's interesting. Let's assume that we take the latest published version of the screening of the genome. There were 32 variants. We call them SNPs. And at each of these SNPs, we have two versions, two alleles that we call them. One comes from your parent, from your mother, and the other from your father. And each one, you can ask whether it is related to positively to obesity or not. If you take the sum of these positively associated SNPs, since we have 32, the maximum score that you can have is 64. So you can find people in the range of 0 obesity favoring alleles to 64. And if we compare those who have a few alleles here, in this particular population of 8,000 people, the low numbers were in the low 20s, and those who had more alleles, it was closer to 40. You see that there is a BMI difference of about 3.5 units of BMI. So it's not a huge effect. It's not 10 BMI units, not the difference between normal weight and class 2 obesity. But from a population perspective, 3.5 BMI unit is not benign. It's like a few millimeters of mercury in blood pressure. We know it makes a huge difference for a given individual when you see this as a population level. Now let me show you how a genotype can impact a phenotype. I'm going to use, to illustrate this, birth weight. We can have maternal or fetal genes that impact on birth weight. And of course, this is a very incomplete picture, but you're going to see how these things come together. For example, this gene, HNF1B, I'm not going to go into the details, time will not allow me to do that. If you have the wrong version of that gene, you have 2 copies of it, the baby will be about 900 grams lighter. So that's almost 2 pounds. This gene, HNF4A, 2 copies of it, you have 790 grams more than the average. The next one, JCK, glucokinase. If you are homozygote for another version of the gene, you are 550 grams lighter at birth. But if the mother has this genotype as opposed to the baby, then the baby will be 600 grams heavier. The reason is that it is a gene that regulates blood glucose, and the insulinic response of the baby will vary depending on the genotype of the mother and cause a weight gain in the mother and a weight loss in the child. These 2 genes, if you have the rare alleles, you're homozygote for them at both genes, the baby weighs 113 grams less. A mitochondrial carrier gene, if you have 2 copies of it, 26 grams less. The FTO gene, the well-known FTO genes, 22 grams only. And in the mother, corticotrophin-releasing hormone-binding protein, 148 grams less. So as you can see, this is a distributed regulation. Sometimes you have a gene pulling a little bit in one direction, another compensate, and the end product depends on the very complex interactions between multiple genes. That's the genotype of human obesity. So my conclusion, observational data suggests that there is a significant genetic component to obesity. There are significant genetic components also to intermediate traits causally related to body weight and adiposity. I didn't talk about the rare variants because I realized that someone's going to talk about it next. There are about 100 DNA variants, all with small effects, have been associated with obesity. So in other words, we're talking about a genetic predisposition, not a genetic determinism. And finally, both observational and experimental data strongly suggest that the response to manipulation of the energy balance, to the physics components of the reality of energy balance, such as caloric restriction, reduced sugar and reduced dietary fat, reduction of sedentary behavior, or increase in physical activity level, or overfeeding, the response is conditioned by genomic variants. And that's not a small effect, it's a large effect size. But the mechanisms are unknown at this time. Thank you very much for your attention. A deadly silence. Roger.
If I interpret the data right, your twin study seemed to indicate that the response to, the response to overfeeding was highly independent of genetics, whereas the response to underfeeding was more dependent on the genetic relatedness of twins. Is that correct?
Well, we had an intra-pair correlation of 0.55 in the overfeeding, and I think it was above 7 in the negative energy balance. So given the sample size, these studies are so complex, you cannot have large sample sizes. Given the low statistical power around these numbers, I think they're probably comparable. But we know that human beings, like all mammals, are much better at defending the body against weight loss. The physiology is geared towards preventing weight loss. The our way of protecting us against weight gain is not very good, as shown by the current obesity epidemic.
Zehawk, Mississippi State, backbencher again. Very interesting talk right here at the very back. Okay. The wonderful compilation of data that you've shown us, but I think you emphasized quite a bit on epigenomics, histone modification, methylation, and so on and so forth. That's obviously the reason why the twins probably varied, the A twin versus the B brother. Because one of them, the B, I think, was prone more to weight gain than the other was. Perhaps it's because he hung out with friends who had bad habits, meaning more eating, need to eat more perhaps, or maybe some kind of environmental factors there. I've seen the Japanese, I've been in Japan many a times, that they are not necessarily eating less, and yet they are so thin. There might be some kind of a dietary impact as well. Have you come across something like that as to how the diet will impact epigenomics and thus impact obesity?
So there's a lot of feedback here. I'm not sure I understood the fine details of the question. I'm not necessarily, I would not necessarily impute the within-pair variance to epigenetics. It could, but it remains to be demonstrated. There are lots of factors that could come into play here. But that's one potential explanation indeed. I'm comfortable with this. What else? I could not hear the end of your talk, so I missed the last part of the question.
I'll just ask you a question instead. So my question is, if you look at your twins at either end of the spectrum, if you look at their genetic variants, does that help you get at what's causing diversity, because they're relatively more homogeneous gene population when you do that? So if you had twins that were at the low end of weight gain versus twins were at the high end, so can you use those, or is that an approach, or is it too limited because the population size is so small?
We have tried to find variants that would discriminate between the high gainers and low gainers, and it's hopeless. When we deal with 50 variants and more, it's so much noise that. But that's the idea. I wish we could do it with 1,000 pairs, and yes, then we could sort that out. Excellent question. Okay, I guess thank you.